A PCR product was ligated to a PCR 2.1 topovector and transformed into chemio-competent and electrocompetent E. coli DH5A cells. The vector contains an ampicillin-resistant gene and a laxida gene, so the cells were cultured in an LB medium petri dishes with ampicillin and ex-gal. However, no isolated colonies were grown, and no blue cells were obtained, only white. This was probably due to the ampicillin and ex-gal used being inactive because of the storage time and conditions.
Topocloning systems use a topoisomerase enzyme that is able to both cut the DNA strands and anneal them later. First, the desired fragment is amplified using a low-fidelity polymerase that will add adenines to the 3' ends of the product. Then, the amplified product is incubated with the topoisomerase, which contains the CCC-TT sequence, which the enzyme will cut, leaving free thymines. Then, the product will be hybridized with these thymines using the adenine overhangs, and the topoisomerase will anneal it. The vector used was the PCR 2.1 topo, which is used for several applications, among which are the cloning of products using this topoisomerase system, the in vitro transcription and sequencing of the cloned genes, and the screening of colonies with the white-blue system. There are several other vectors that can be used with the topoisomerase system. For example, the PCR BLOND2 topo has the LAC-C and the CCDB sequence with the recognition site for the topoisomerase. This sequence produces a toxic compound for the bacteria, and this allows us to select the colonies that were transformed with the ligated product. It also contains the resistant cassettes for ampicillin and kanamycin, and it is useful for the cloning of products with blunt ends, so it allows for the use of a polymerase that has a high fidelity. Another example is the PET 100 directional topo. This contains the cassettes for ampicillin and kanamycin resistance, as well as T7 promoter and a histidine and epitope tag. It also has the recognition site with one blunt end and one sticky end, so it allows to incorporate the product in a directional manner and then express it. Finally, the PBAT topo vector contains the resistance cassette for ampicillin and kanamycin, as well as the recognition site for topoisomerase and the PBAT promoter, as well as the RAC gene. So this is useful for the expression of the cloned genes with the topoisomerase system with an arabinose inducible manner. For topocloning into competent E. coli, sodium chloride and magnesium chloride should be used in the ligation reaction. However, when using electrocompetent E. coli, the salt solution should be diluted to prevent arsing of the cells. In this way, the salt solution must be diluted 4 times to ensure a proper ligation solution.
The PCR product for a topo ligation with this vector should have a 3' adenine overhanging, so it can hybridize with the free thymines in the vector. To achieve this, the PCR amplification should be performed with a low-fidelity polymerase, such as Taq DNA polymerase. The cloning selection system with ex-gal is based on the fact that the laxida gene codes for the beta-galactosidase, which when reacted with ex-gal provides blue coloration. This gene is located in the plasmid at the center of the cloning site. So if an external sequence is inserted in this part, the gene will be inactivated and will not produce beta-galactosidase, so the cells will grow white. On the contrary, if nothing is inserted, the gene will remain intact and the colonies will be blue. Some ways to verify that the colonies obtained have the desired insert are sequencing using the primers M13 forward and reverse that are contained in the multiple cloning site of the plasmid, to sequence, deconstruct, and confirm the presence of the insert and its correct orientation by comparing it to the original sequence. Also by PCR using only one of these primers M13, forward or reverse, and one primer that hybridizes with the insert. Then analyze this in an electrophoresis gel and compare the size of the bands with and without the insert. Also, it can be done by restriction analysis. It first is needed to culture from 2 to 6 colonies of interest, white or blue, overnight in LB medium with antibiotic, ampicillin or kanamycin. Then isolate the plasmid with a pure link quick plasmid microprep kit and use restriction enzyme or a combination of restriction enzymes that cut the vector and the insert to confirm the presence and the correct orientation of the insert. The PCR 2.1 topo plasmid has a lot of elements that have different functions. For example, the OREF1, it's an origin of replication for single-stranded replication and packaging into phage particles. OREPUG, it's an origin of replication for high copy number and maintenance of the plasmid in E. coli. Ampicillin and kanamycin restriction sites, genes, are for selection of E. coli. The laxida gene, it's a gene for blue or white colony screening and selection of the recombinants. The T7 promoter, it's for in vitro transcription. The M13 primer sites are for in vitro sequencing or PCR, and it also have a lot of restriction sites. For example, ECOR1, which is a site flanking the PCR product insertion, site for easy excision of the insert.
It has been made a PCR product ligation in vector PCR 2.1 topo of in vitro genes, which contains topoisomerase to do the ligation of the PCR product, with adenine ends as other elements that promote high replication. This was made in E. coli, DH5-alpha that has necessary characteristics to express the plasmid. It is observed in Figure 1 and 2 the control and ligation done in the laboratory. Figure 1 shows growth of bacterial colonies color white with any presence of blue colonies. The number of colonies is uncountable, so the efficiency of transformation was not obtained. Figure 1. Transform E. coli DH5-alpha, group with plasmid 2.1 topo with a ligation of PCR product in medium LB, ampicillin and X-col. Figure 2. Positive control with E. coli DH5-alpha without transformation in medium LB with ampicillin and X-col. Since in control blue colonies must be grown, it may be an indicator that X-col didn't work properly, so it is recommended use reactant previously tested to check its operation.
Vector PCR 2.1 topo is a cloning vector. It acts as a transfer molecule for DNA inserted with a recombinant technique. By using topoisomerase 1, we can cut the plasmid on this 5' 3-cytosine to thymine zone, leaving free thymines that covalently bond to the topoisomerase on the tyrosine 274. This mechanism allows that the PCR products inserted with adenine together releasing the topoisomerase and creating a vector with a fragment of DNA inserted.
To perform a ligation, it's essential to know the vector's characteristics and elements because that's the way to learn if it has any gene that helps identify the ligation procedure was successful. If it has antibiotic resistance, among other benefits that help prepare correctly the cultural medium and determine the growth conditions. Some of the most important elements for the PCR 2.1 topo are like set fragment, M13 reverse priming site, multiple cloning site, T7 promoter, M13 forward priming site, F1 origin, PUC origin, kanamycin and ampicillin resistant ORF.
Other vectors in the topo family used for insertion of PCR products, but in this case with blunt ends, just like the PCR blunt 2 topo. which is a vector designed to clone PCR products with blunt ends. These products are generated by a thermostable proof-reading polymerase, such as platinum superfine DNA polymerase. It has a high fidelity level, extensive 3 to 5 end exonuclease activity, and do not leave adenine rings, so the PCR products end up blunt. The insertion follows the same process as PCR 2.1 topo vector, and the other components of the vector are the same, just as shown in the following images. When working with these vectors, it's important to verify if the DNA has been inserted into the vector. For this, there are different methods, including common PCR, enzymatic restriction, and sequencing. For conventional PCR, we can take advantage of the primers that are already inserted in the vector, which are M13. These are well-designed and studied primers, which can be easily amplified to verify that the DNA inserted is already there. We can run this at the agarose gel and perform the normal PCR. On the other hand, a diagnostic restriction enzyme, digest, takes advantage on the fact that the restriction enzymes leave DNA clipped at specific sequences, called restriction sites. Often, the size of the plasmid insert and vector backbone are known, and thus this technique can be quickly used to verify our plasmid. The goal of the diagnostic digest is to cut the plasmid into specific sized pieces and analyze the resulting fragments by gel electrophoresis. The pattern of the fragments on the gel can indicate if the plasmid contains the expected size fragment. By selecting the appropriate enzymes, we can either linearize the plasmid to determine the entire size of the construct or excise some or even all of these insert from it. Finally, sequencing is such a specific and necessary process to verify not only the insertion of the plasmid, but also the direction of the insertion and even possible mutations. In this process, the same primers that the vector has are used to determine the order of the base pairs that are contained in the DNA insert.
A method that commonly used in order to identify recombinant bacteria is the blue-white screening, in which the growth of a white colony means a successful recombination, while a blue colony shows that the plasmid doesn't have the insert. If the color change in the colonies is 100%, it's not reliable because sometimes small sequences of DNA may be inserted into the multiple cloning sites and change the reading frame of ligation gene, resulting in false positive wild colonies. Mandar esto. Ah, no, se está reanudando. An important factor in the transformation and that has an impact on the efficiency is the bacteria strain use. The E. coli DH5 alpha strain are cells that reach high transformation efficiency, and some of its characteristics are activity of non-specific endonuclease 1 and A1 eliminated for highest quality plasmid preparations, resistance to phage T1, blue-white screening, reduced recombination of clone DNA, REC-A1, and efficient transformation of unmethylated DNA derived from PCR, cDNA, and many other sources.
To obtain a successful ligation, it is necessary to have a PCR product with a high degree of purity, as well as efficient competent cells and a good plasmid design. The obtained colonies were considered countless, so the efficiency was not calculated.